there, kids! Welcome to today's talk. I'm Justin,
I'm Remy.
and we are here to talk to you today about the Tensegrity model of the cell. We're going to be telling you about how it all works, how the cytoskeleton functions, and what it can be used for. Isn't that right?
Exactly.
Oh wait, did we forget someone?
I think we did.
Luke,
Hey there.
go fish.
You know, Mr. Bones, I was looking at our cells under the microscope today, and they just such a variety of shapes and sizes. But I gotta ask, how do cells maintain those structures?
I'm glad you asked. Hello there, Remy.
Where did you come from?
Yeah, never mind that. Anyways, the cell. It's an amazing thing.
Yeah.
Oh, why do they maintain the shape? Why aren't they just squished around? Well, here you have a cell. Nice round cell right there. And a cell has a lot of different kinds of fibers running through its body to maintain its shape, maintain organization of organelles, and even allow it to move. So you got things like the nucleus here in the center. You got some random organelles along the line here. And in a cell, you have three main fiber types. You have microtubules, you have actin fibers, and you have intermediate filaments.
Now, Justin, where have I heard of actin fibers before?
I'm glad you asked. So you may have heard actin fibers because these are the same proteins that are, along with myosin, responsible for allowing your muscles to move. And in a cell, they serve a very similar purpose. You see, the bottom of your cell it's covered, inside your cell is covered with actin meshes. And these meshes can grow and subtract. And by doing so, they can allow the cell to change its shape or even allow extensions to grow. Things that we like to call pseudopodia. Microtubules, on the other hand, are responsible not for the shape of the cell, but for organizing the cell itself. So we have these little things on the inside called centrosomes, and they are the origin points of these microtubules. And what they do is that the microtubules allow the organelles to attach themselves and spread out throughout the cell. And in this way, the cell has a degree of order to them. And finally, the intermediate fibers serve different purposes throughout all different cells and are responsible for many different purposes, but it's unique by cell type. So, the cell.
The cell.
The cell.
What about that, Mr. Bones?
So how does the physics behind it all work?
Excellent question, Mr. Bones. I'd love to explain. You see, the key concept is tensegrity, or tensional integrity of the cell. It works with compression and tension so that things are kept together but aren't squeezed like a balloon. I'd be great if I have a model for this. Oh, wonderful. This is a truss and strut model. It pictures it like elastic rubber bands and compressive steel rods which keep the shell together. Here, I've pictured one on the board. The physics behind it works by considering one point on the truss and the different forces and tension at play. Here, I have four forces. One measuring 4 times the square root of 2 newtons, one measuring 4 newtons, and two more measuring 4 newtons, but they're pulling at different angles so that when you resolve each, you get different resolutions, and then you can add them all together. Now, you add them all along their respective axes, z, y, and x. The x-axis adds the two forces from here, which equals 0. Now, the compressive force from here has a j and a k component, which are canceled out respectively by the forces in these two elements under tension. And that's how the physics works, Mr. Bones.
Now, this piece really speaks to me. I call this one amoeboid lunch. Funny enough, it's actually got a lot of similarities with how I get my own lunch. Movement itself is fundamental to survival. Humans move to run from predators, to eat, to express emotions, and do a variety of important tasks that we cannot function without. Not only humans, but all animals and the incalculable amount of microscopic organisms living around the world require movement to survive. To achieve this, humans and most animals have muscle cells and permanent appendages filled with said cells that allow us to move around varying terrain and obstacles. So, how do single-celled organisms accomplish this task? Pseudopodia. Microorganisms create temporary arms and legs by pushing on one section of their cytoskeleton, held together by tensegrity, and extend the cell membrane out in the desired direction for all their appendage applications. Actin filaments relax in the region to facilitate this change in shape. The cytoplasm filling the cell moves into the newly formed appendage to complete this process. Once the microorganism is finished with its pseudopodia, their cytoskeleton tensegrity returns to normal and the appendage collapses into the body. Isn't that neat? Now I think I might as well make some pseudopodia and get my dinner now.
Can I have some dinner too?
Sure, if you can create your pseudopodia.
Alright, well, this is the end of the video. Hopefully you've learned a lot of great stuff. So to recap, our cell has three main types of fibers. We got the microtubule fibers, we got the actin fibers, and we have the intermediate fibers. The actin fibers are related to movement and to keeping the shape of the cell.
I hope you remember that tensional integrity is the characteristic property by which cable and strut elements keep the cell in tension and compression to maintain its integrity.
And finally, these principles combined create the foundation for how we live and every different organism under the sun. Thank you so much for watching. And frankly, I've got a hankering for dinner.
Me too.
Me too.
Let's go.
Let's go.
What about my dinner? Woo-hoo-hoo-hoo.